Tropism is defined as growth or movement of a plant in response to environmental stimuli. One type of tropism, known as phototropism, is the growth of a plant in response to light. As you can see, when a light source is added, the plant begins to reposition itself by bending towards the light. We now know that plants move in this way towards light to maximize photosynthetic activity in order to optimize growth and health. But how does this movement occur? Charles Darwin studied the phototropic response, which controlled the direction of organ growth within the plant, linking to follow the direction of the light source. Darwin proposed that there was some matter in the upper part of the plant, which is in turn being acted on by light, hence transmitting its effects to the lower part. The photoreceptor proteins which are responsible for phototropism are called phototropins. Phototropins detect incoming wavelengths, specifically blue wavelengths emitted by light. Important to phototropism is the plant phytohormone auxin, which is the central regulator of growth and cell elongation in plants. The light received by phototropins causes a chemical reaction that establishes an auxin gradient in the plant. In the shoot, the auxin develops and stimulates cell growth and, more importantly, cell elongation on the side of the plant farthest away from the light source, causing the hypocotyl, which is in the stem, to bend in the direction of the more light-exposed part of the plant. This growth towards the light source is called a positive response. This new position allows more light to reach more cells on the plant, which are responsible for photosynthesis. Since plants are constantly growing, this auxin-mediated directional growth response can alter the orientation of plant growth quite rapidly. The plants on the far left did not reorientate themselves towards the light source, as the apex, which is the source of the auxin and the location of the phototropins, has been cut. The next group of plants had the apex covered with foil and did not reorientate towards the light source either. The other group of plants had the top covered by clear wrap. As you can see, they reorientated themselves towards the light source. This is simply a control to show that whatever is used to cover the stem must not be clear. Next to this group are plants with foil wrapped around the base and the apex. As they still reorientated towards the light, despite the foil, this proves that phototropins are in the tip. The last group of plants are another control group and have not been altered in any way. Plants are in fact very specific as to what wavelength they absorb in various processes. Phototropin proteins 1 and 2 specifically recognize and respond to only wavelengths in the UV blue side of the spectrum, which is approximately 450 nanometers, which triggers hypocotyl curvature. This experiment shows that when exposed only to blue light, the plants still reorient to the light source. Now, we can see what happens when the plants are only exposed to red light. As you can see, the plants do not bend towards the red light at all. Finally, When exposed to red light on one side and blue light on the other, the plants clearly bend towards the blue light, thus showing that the plants bend specifically towards blue light and not the red. Hence, the plant responds to the UV blue wavelengths for phototropism, indicating that it is the blue light perception and signal transduction and response mechanism that drive phototropic curvature of the stem. Since Darwin proposed that there was some matter in the upper part of the plant which is being acted on by light, 5 hypothetical models for the way an auxin gradient is established have been made for the model plant Arabidopsis. In the first model, incoming light deactivates auxin on the light side of the plant, allowing the shaded part to continue growing and eventually bend the plant over towards the light source. The second model proposes that light inhibits auxin biosynthesis on the light side of the plant, thus decreasing the concentration of auxin relative to the unaffected shaded side. The third model states that whilst there is a horizontal flow of auxin from both sides of the plant, incoming light causes more auxin to flow from the exposed side to the shaded side, increasing the concentration and thus growth. The fourth model shows receiving light to inhibit auxin basipetal or vertical transport down to the exposed side, causing the auxin to only flow down the shaded side. Finally, Model 5 is a bit of both Model 3 and 4. The main auxin flow in this model comes from the top of the plant, vertically down towards the base of the plant, with some of the auxin travelling horizontally from the main auxin flow to both sides of the plant. So, in this case, receiving light inhibits the horizontal auxin flow from the main vertical auxin flow to the irradiated, exposed side. As shown in the animation, plants bend in the middle of the stem to reorientate themselves. So, is this where the photoreceptors are found? Actually, phototropids are located towards the top of the plant, in the apical meristem, and so the light is perceived there. The signal is then translated into an auxin gradient at the point of bending. This is an example of translocated signal response mechanism. Bending back from the tip has the potential to increase the exposure to light more rapidly because the tip can be moved substantially towards the light. In essence, phototropins respond to light sources that they trigger auxin flow to cause cell growth and elongation on the shaded side, reorientating the plant towards the light source. The plant does this in order to attain maximum photosynthetic activity when necessary. From the Darwin experiments, it was clear that the phototropins are located in the tip or apical meristem of the plant and are the center for auxin signaling. Furthermore, these photoreceptors specifically recognize and respond to UV blue wavelengths as shown in the blue light, red light experiment. Phototropism is really an important process for health and development of plants throughout the world.